Hi, I'm Spencer Krieger, and I'm excited to share this work today that I did with my PhD advisor, John Kececioglu, titled Fast Approximate Shortest Hyperpaths for Inferring Pathways in Cell Signaling Hypergraphs. Cell signaling is how cells exchange information with their environment by sending and receiving small molecules at membrane-bound receptors, which is fundamental to cell biology. A cell signaling network encompasses all of the chemical and physical reactions that occur in the cell. A cell signaling pathway is a subnetwork of these reactions that describes a cellular process and usually begins at one or more membrane-bound receptors and ends at transcription factors and regulators. Here, I show an example of the p53 signaling pathway, which is involved in DNA repair and apoptosis. Shown on the left, when a stress signal is sent, p53 promotes DNA repair and downstream apoptosis by aiding in the transcription of several proteins. However, shown on the right, when there is no stress signal, it is inactivated and is degraded by MDM2. Perturbations in this p53 pathway causes cancer. Notice the series of reactions when p53 is deactivated by MDM2, which starts at p53 in the cell and ends with p53 and MDM2 being transported from the cell as the target reaction. The reactions that make up these signaling pathways have this general form, with the reactants, here A through B, on the left, and the products, here C through D, on the right. The reaction will only proceed when all reactants on the left are present, and when it does, all products on the right are produced. Inferring the most likely series of reactions or pathway from a given set of receptors or sources to a given transcription factor or target is a key task. We use directed hypergraphs, which are a generalization of ordinary graphs, to correctly model these cell signaling pathways. A directed hypergraph consists of vertices and directed hyperedges, where a hyperedge generalizes an ordinary graph edge and is a pair of vertex subsets x and y, where now the edge goes from a set of vertices, its tail, to a set of vertices, its head, as shown here in the picture. To traverse this edge E, all of the vertices in the tail must already be reached, and upon traversing the edge, all vertices in the head are reached, like in the reaction I showed on the previous slide. In a cell signaling hypergraph, vertices represent molecules, including proteins, and hyperedges represent reactions. A specific signaling pathway is a series of hyperedges from source vertices to target vertices, which is formalized as a source-sink hyperpath. In this picture, this hyperpath goes from S on the left to T on the right. Notice that there can now be multiple outedges from S, as shown on the left. In the middle, hyperedge E highlights a general hyperedge, where all of its tail vertices must be reached by prior edges in the hyperpath for it to be traversed. And T on the right is one of the vertices in the final hyperedge. The problem we are trying to solve is the shortest hyperpaths problem, which is, given a hypergraph, positive edge weight function omega, source vertex S, and sink vertex T, find an ST hyperpath of minimum total weight. This finds the most likely cell signaling pathway between the source and the target. The current state of the art for shortest hyperpaths in cell signaling networks finds only acyclic hyperpaths, and it does this by solving a mixed integer linear program, or MILP. Shortest hyperpaths is NP-complete, and because the MILP solves this NP-complete problem, it does not have any efficiency guarantees, but does do well in practice. Feedback cycles are ubiquitous in signaling networks, and different from ordinary graphs, shortest hyperpaths can contain cycles, as shown here in the picture, where this ST hyperpath contains a cycle, shown in red, by the single hyperedge which has a previous vertex from the hyperpath in its head. In the common case where all hyperpaths from S to T are cyclic, the MILP finds no solution, even though ST hyperpaths exist. We give the first efficient heuristic for general shortest hyperpaths that can contain cycles and runs in low-order polynomial time in the worst case. We also give a simple and practical hyperpath enumeration algorithm, which allows us to tractably measure how close our heuristic is to optimal. Experimentally, our heuristic matches the state of the art on over 99% of all acyclic instances from 2 standard cell signaling databases. And when every hyperpath is cyclic, the MILP finds no solution, so our heuristic surpasses the state of the art on cyclic instances, and it was shown to be optimal on all such instances as verified by our hyperpath enumeration algorithm. To preview our shortest hyperpath heuristic, I will start by introducing Dijkstra's shortest path algorithm for ordinary graphs, which will then be contrasted to our heuristic, which proceeds in a similar manner but with a few key differences. Dijkstra's algorithm finds the shortest ST path in ordinary graphs. It maintains a heap of vertices prioritized by their estimated distance from the source as their key, and each vertex holds on to its best in-edge. The heap is initialized to the source vertex S, and until it is empty, we repeatedly extract the vertex V with the smallest key, which is the length of the shortest SV path. Then, for each outedge of V, edge VW, we update W's key with the length of the best SW path we have so far. And then we hold on to the best in-edge to W. Finally, we recover the shortest ST path starting from T using the best in-edges stored in step 3. Similarly, our heuristic finds an approximate shortest ST hyperpath. The heuristic also maintains a heap, but is a heap of hyperedges instead of vertices, with an edge's tail distance as the key, which is the length of the best known hyperpath from the source to the vertices in the tail of E. Here we keep a list of in-edges, as a hyperpath ending at a tail set consists of many final hyperedges. We initialize the heap to the hyperedges with the source in their tail, and repeatedly extract the hyperedge with smallest key, which is the smallest estimated tail distance. And for each outedge F of edge E, we update F's in-edge list with E. We then use a greedy heuristic to approximate the shortest hyperpath from S to the tail of F, which is also NP-complete, and update F's key if this approximate shortest SF hyperpath is shorter than previous ones. Then, we again use this greedy heuristic to recover an approximate shortest ST hyperpath. To determine the quality of our heuristic in practice, we constructed hypergraphs using human-curated signaling pathways from standard pathway databases, NCIPID and Reactome. We map each entity, be it a protein or small molecule, to a vertex, and each reaction to a hyperedge. Any vertices with no in-edges we use as sources, and any vertices with no out-edges we use as targets. For each target instance, we will use all of the sources, but we are only trying to find a hyperpath to one target at a time. I will now share the results on the acyclic instances from our hypergraphs, which are instances where there is an acyclic ST hyperpath. Of the reachable target instances for each dataset, most are acyclic, as seen when you compare the top and bottom numbers of each column. On over 99% of these instances, our heuristic matches the state-of-the-art MILP, while providing a guarantee of an efficient running time, which is not provided by the MILP. In cyclic instances, where all ST hyperpaths are cyclic, the MILP returns no solution because no acyclic ST hyperpath exists. Looking again at the reachable target instances, we see that there are many cyclic instances. In all of these cyclic target instances, our heuristic is optimal, which we proved by enumerating all hyperpaths, choosing the shortest one, and comparing its path length to the heuristic. I will now highlight one of these cyclic instances so you can get a feel of what these shortest hyperpaths look like. Here we show the shortest hyperpath to the phosphorylated p53 tetramer, which shows the activation of p53 in the cell. Here, the sources are along the top of the figure, which are vertices in the hypergraph with no in-edges, so we assume that they are already present in the cell. The target is on the right, the phosphorylated p53 tetramer. Edges E2 and E3 are dashed because they show transcription of a protein, and the small circle with a P inside shows phosphorylation. So here, transcription factors FOXO3 and FOXO4 facilitate the transcription of STK11 in the hyperedges E1 and E2. P53 is transcribed and forms a homotetramer in the hyperedges E3 and E4. This tetramer then forms a complex with ARK5 and STK11 to allow its phosphorylation in E5. Then ARK5 is phosphorylated in E6, and P53 is phosphorylated in E7, which breaks the complex and causes a cycle recreating STK11, which was already reached in the hyperpath. In this talk, I have presented the first heuristic for the general shortest hyperpaths problem. that properly handles cycles instead of only allowing acyclic hyperpaths, is guaranteed to be efficient instead of having possibly exponential running time, that matches the state-of-the-art MILP on 99% of all acyclic instances, and that surpasses the state-of-the-art on all cyclic instances, where the state-of-the-art returns no solution, and we have shown that the heuristics hyperpath was optimal. I'd like to thank T.M. Morali and Anna Ritz for their helpful discussions about this work and the U.S. National Science Foundation for funding. I'm a PhD student who will be graduating in the spring of this upcoming academic year, and I am interested in postdoc positions. Thank you for listening to my presentation.